Thank you. Okay, thank you. I just open my window. Is it visible? Now? Okay, just wait. Is it visible? It is okay? Is it okay? If it is visible? If it is visible or not? It is not visible, but I am sharing. Just wait. Is it okay? This one? I'm trying. PPT is visible or not? Some error. Okay, just wait. I'm trying. Now is it is okay?
Now it is visible.
Visible?
Yeah.
Yes.
Go to the slides there.
Yes. Is it okay? Can I start? No, this is slide share view is there. I also open it.
Five, whatever you like.
Is it visible?
Screen is visible, but it is not in the slide share mode.
No, I am also sharing is slide share view.
F5, press F5.
Yes, F5. In my
F5
case,
will.
it is also in white full presentation view. Yes, this is the initial side, and I am just going through next slide like that.
Okay, right side, go to the right side. Sorry, go to the right side, then click the slide view. The
Yes, right side.
percentage sign, this click this one. Okay, this one.
Yes.
Oh,
Is it
yeah.
okay?
Now it is. Now it is okay.
Okay. Thank you very much. The organizing committee from Department of Biotechnology, Haldia Institute of Technology, for providing me this opportunity to present the research work related to computational drug discovery and molecular docking techniques. Today, I will present a few research work related to drug discovery of different proteins. The title of my work is Role of Conserved Water Molecules in Computational Drug Discovery. I am presently working as an assistant professor in Department of Bioinformatics and Computational Drug Discovery and Biomolecular Simulation Lab, which I established at Department of Bioinformatics, Macau. Before starting the computational drug discovery part, I would like to thank you, the Nobel Prize winner of 2013, the three-person, Professor Martin Karplas, Michael Levitt, and Aris Wurzels. As per their informations, in the real world, this could be eventually mean that most chemical experiments are conducted inside the in silicon of chips instead of the glassware or laboratories. Turn off that Bunsen burner, it will not to wanted in 10 years. So that means before going to perform any experiment, we have to do some computational work or in silico work. So that will be just less the time and also the other part. The entire talk and entire research work I have divided in two parts. One for CML cancer and another for COVID disease. Okay, so these are the three subtopics: identification of conserved water molecules and prediction of their dynamical function, water-based drug discovery, and mechanism-based drug discovery. So drug discovery, as we know, there is different technique is there, like target identification, validation, high throughput screening, like that. This is the common general area for drug discovery. So a few work today I will present, like structural, functional, and dynamical role of conserved water molecules in the ligand binding sites. The work is cited in the front page of the journal from American Chemical Society 2021. Me and my few students from Central University Jamia Millia Islamia performed this work. So identification of conserved water
Sir, kindly
molecules.
can I interrupt you, sir?
Yes.
Sir, your slide is not in full screen mode. That's why
It's not
it is very small and to see it very problem.
so, but in my cases, I am seeing it.
So you, sir, you go to the those button, home, insert, design, animation, and again you see that is slideshow. In the
Slideshow.
presentation,
Yes.
PPT,
Yes.
slideshow, you click slideshow and then from beginning.
From beginning. It is okay.
Then click from beginning.
Yes, I have clicked it. Is it okay?
No, it is not. Not actually.
Yes. In slideshow and from beginning, I am also pressed is here one.
Yes.
This
Slideshow
one.
from beginning.
Yes, slideshow from beginning. This one.
Yes.
It is visible to everyone.
No, it is very small
Sir, one
actually.
thing can I say? Can you just reopen your slide once again?
You close and
Problem
again
is
open
happening.
again.
Okay.
You close the entire.
Or else you after opening you just press F5. F5 key.
Is it visible this folder?
Yes.
Yes, sir.
Now.
Now you press F5.
Now
F5.
you.
Yes, F5. I press it. In my case, it is also
Okay.
Yes,
visible
sir.
to me.
Now
Yes, sir.
it is
Yes, sir.
okay.
Now it is
Now it is
totally
okay,
okay.
sir.
This
Okay.
one okay?
Yes, sir. Yes, sir.
Yes. Okay.
Okay. Thank you, sir.
Okay.
Okay, thank you, sir.
Okay. So this is the title of the work: structural, functional, and dynamical role of water molecules in ligand binding site of the human GMPR enzyme. The work is published from American Chemical Society in the front page of the journal in 2021. Identification of the conserved water molecules and prediction of their dynamical function. It is very important. The work I have started from 2006. Now work is going on. So the basic problem is that the two protein IMPDH and GMP reductase are mainly responsible for leukemia cancers. Okay, so the two cell, as you observed here, normal cell, left side, and right side leukemia cell. So IMPDH, GMP synthetase, and GMP reductase, all these three enzymes basically control the structural and functional integrity of the systems. That basically purine nucleotide biosynthesis pathway. So this schematic presentation I have prepared from my research work. So IMPDH, GMP reductase, there is two binding pocket, mononucleotide binding and dinucleotide binding. The mononucleotide binding sites, the substrate is stereospecifically bind to this position. Then the cofactor NAD will come and bind to this position, and then some chemical reaction will be there, and the product will be released from XMP. From XMP, it will take by another enzyme, GMP synthetase, and then GMP synthetase release the GMP, which will be uptake by the another enzyme, GMP reductase. So these are the basic background of the research work. This is basically conversion from IMP to XMP to GMP. The entire structures I am presenting here: GMP reductase, GMP synthetase, and IMPDH, like that. In this way, they will control these systems. The IMPDH and GMP reductase both have two isoforms, isoform 1 and isoform 2. Generally, isoform 1 protein is found in normal cell and which act as a housekeeping role, whereas isoform 2 may found in leukemia cells. The basic problem is that the IMPDH1 or GMP reductase 1, that's mean isoform 1, is found in normal cell, and the isoform 2, IMPDH2 or GMP reductase 2, they are found in cancer cell CML. This is the presentations. And these are the 1JC and 2BLE. This is the PDB ID, which is crystallized by and submitted by different crystallographic group. So when we align these two sequences, GMP reductase 1 and 2, you observed their sequence similarity is around 90%. Here I am presenting this position, the green color, almost green color, they are sequence similarity, and few position, the red colors, they are dissimilar region. When I superimpose both the structure of GMP reductase 1 and 2, that's mean normal protein and cancer protein, you can see here the middle region, they are almost structurally similar. So if we design some drug or some ligand or inhibitor, it may bind both the position GMP reductase 1 and 2, so it will kill the normal cell also. So that is the main problem for computational drug discovery. So this is nowadays the scientists are thinking to provide some new dynamical model so that the isoform 1 and isoform 2 should be distinguished. These are the inhibitor of CML cancer like ribavirin, maziripin, mycophenolic mofetyl, like that. But they kill also the normal cell. That is the main problem. So when we take the structure, isoform 1 and isoform 2, we observe the GMP, basically the substrate, and the product IMP. Here you can observe IMP and GMP, their sequence and their structural integrity is almost similar. The biochemical mechanism of this GMP reductase enzyme is that they take the substrate IMP, then cysteine 186 will be there, and some chemical reaction will be processed. They produce the product IMP. So this is their biochemical mechanism. So here I am thinking, if I produce this entire mechanism by molecular modeling and MD simulation techniques, so what will be happen next? These are their structural position of GMP and IMP and the interaction with the residue in X-ray structures. So what I am planning, I prepared the both model structure, that means GMP reductase 1 and 2, isoform 1 and 2 in 4 subtype. One for substrate bound conformation, GMP. Next, middle region, substrate with cofactor. Third, product with cofactor, then product. So the enzymatic mechanism will be followed from substrate to product in initially, and middle part will be your intermediate state. So in this way, we have modeled these two protein, and run the all atom MD simulation up to 100 nanosecond. I will check the two model, what will be happen. Then what we done, we identify the water sites. Basically, the water molecules, if the water molecules between the two structures it is within the 1.8 angstrom, we'll think that will be conserved in both the structures. This is the actual nature for identify the conservedness of a molecules. So here we are thinking the water clusters, and these are thinking about the hydration sites. So within these hydration sites, if one water cluster is there, and they will definitely bind to any residue or ligand with hydrogen bonding distances, they will be think as a conserved water sites. Then we calculate the inhomogeneous fluid solvation theory. As we know, the del G is equal to del E minus T into del S. This is the general formula. As we know, the inhomogeneous fluid solvation theory, which is predicted by Lazaridis et al. In 1999. So as per this theory, we produce this equation, del E, this is the energy differences, WP, that's mean interaction energy between water with hydration site of protein, plus half into EWW. EWW means mean interaction energy of water with other hydration site of water, and then minus EW with bulk water molecule. So this is the basically equation which I retrieved from Lazaridis et al. Papers. Then we also calculated their their excess entropy. Excess entropy minus T into del SEX, SR and ST is the rotational entropy and translational entropy. So in this equation, we calculated every water site excess entropy. And also we calculated one water molecule binding energy by value J. J is equal to minus RT integration of WFEP. WFEP means water finding probability. This water finding probability we obtained from the previous equations, and put this value and calculated the binding energy of the particular water molecules. This is the MD simulation study, which you have already seen here. GMP, the first one is GMP, left side, and glutamic acid, threonine, cysteine, they are moving like this way. And then the GMP, like that, this way they are their dynamic behavior. Finally, the left side, this is the intermediate state, and left side you are seeing the product binding position. So all are like this type of dynamic behavior. We just presenting these slides. So what is the enzyme substrate interaction in MD structures? In isoform 1, basically GMP reductase 1, we observed the GMP. Basically, when the GMP is bind in MD simulation, the serine 288 in the red mark, they are far away. But when the NAD will come and bind to this position, that means substrate with cofactor, the far away serine 288, it comes and close to GMP binding sites. That means GMP, and it is also written this position here, here, and here. So in this way, this is this enzyme to product binding mechanism of non-cancerous protein HGMPR1, and second one is the cancerous protein of HGMPR2, which you observed here. The similar way, there is nothing, any changes of or new residues which is available to binding the site. So this is their normal MD structures. But when we calculate the dynamical behavior of normal protein and cancer protein, we observed the cancer protein is more dynamic in nature than the cancer, than the normal cancer protein is more dynamic in nature than the normal protein. This is their dynamics of water molecules. So previous slide, we saw the dynamics of substrate or product. This is the effect of water molecules of that particular positions. So these four conformation is there: substrate bound conformation A, B, substrate with product bound conformation, C, product with cofactor bound conformation, and then D is the product bound conformations. So we observed here W1 at first enter from A conformation to B, and then when B to C, it will depart it from these positions. That is very interesting. And finally, we also observed the W9. W9 enter when the conformation changes from B to C, it will enter and then it will left. So these are the basic interaction, and this interaction is very interesting, which I observed in IMPD GMP reductase 1 dynamics model. This one is the basically GMP reductase 2 dynamic model. We also see when the conformation A to B, the following water molecules come and bind to this position, only W9 is departed from this position, and other water molecules like W2 to W18, they enter these sites. Okay. During the conformation from B to C, the following water molecules like W2, 7, 8, 12, 17, and 18, they departed from this position, and W3 and 19, they are enters. So in this way, we can observe their dynamic behavior. So what will be happen? After MD simulations, these are the tabulated form which we observe: the conformation substrate, substrate with product, substrate with cofactor, then product with cofactor and product bound conformation. This water molecule in normal protein, and this protein, these water molecules in cancerous protein. Fine. Change of conformation, that means substrate to substrate with product. Some water molecule entered the positions like W1 enters, and these water molecules are departed. Similarly, in the case of normal protein, in cancer protein, these water molecule is entered and these water molecule are displaced. So basically, entrance and departed of water molecules is visualized by these slides. So why the water molecules moving from one place to other place, and why the water molecules is appeared or enter to this position? So during dynamics, when we observed that some residue are moving towards the ligand, or some residues is departed from this ligand. This is the main reason for entered and departed of water molecules. Just for example, this is for normal protein, this one is the cancer protein. So A and B both are moving the residue towards the substrate. So this is residue R286, they moving towards the substrate 0.95 angstrom, here also 1.18 angstrom in normal protein. In cancer protein 0.33, 0.9, like that. So they are moving towards the ligand. Okay, that's why due to this movement of residue towards the ligand, the water molecules is departed, like here W4, here W9, here W9, like that. So this is the reason we have find it. Similarly, these residues which are away from the ligands like substrate GMP or substrate INP, that's why water molecules is also moving. Like in these cases, just for example, slide C, these residues which are departed from away from the substrate GMP, so there is one gap and water molecule is departed, like this way, this way, this way, and this way. So this is the basic features of the appearance and departed of water molecules. The work is published in Protein Structure Function Dynamics papers. Then, when we compare the normal protein and cancer protein and their dynamic behavior, we categorize the three parts: exchange, semi-exchange, and retain. What do you mean by exchange? So W1, W4, W5, W6, this green color. Exchange means this hydrophilic position. Within this hydrophilic position, the water molecules are very dynamic and they are departed from these positions randomly. Suppose in W1 position, 10 water molecules during MD simulation we observed 10 water molecules is within W1. Okay, so they are departed, then again come. They departed, again come. So that is the different water ID. They will come, bind to the W1 position, then again departed. Then another water molecules come and bind to this position and departed. So that's why they are highly exchangeable. Okay. Another is semi-exchange, red color. So W9, W13, and W19. They are semi-exchange, means some water molecules departed, step some places, then again return to this position of W9, 13, and 19 positions. That's why we categorized it in semi-exchange. And another is retain. That means these water molecules, this green color, W1, W12, W4, and 15, they are always conserved in all the conformation and only one water ID is there. So in this way, we have categorized these water molecules and we also calculated their interaction energies. When we compare the RMSD value of the normal protein and cancer protein, we find the normal protein only 4 region, 1, 2, 3, 4. As we observe, this region is highly dynamic in nature. Whereas in cancer protein, 1, 2, 3, 4, 5, 6, 7, this 7 region is highly dynamic in nature and their RMSD is approximate 2 angstrom. So that indicates the dynamical model of cancer protein is highly flexible compared to normal proteins. After that, what we have done, we categorize these 19 water molecules into 4 steps. Category 1, 2, 3, and 4. Category 1 means they will find in GMPR1, that means isoform 1, normal protein. Category 2, that is only available in GMPR2, okay? And category 3, they are available either 1 or 2, that is in both, either this one or this one. And GMPR1 and 2, they all are present in all the conformation of these proteins. This one is the basically categorization and their different energy function. Each water molecules we have calculated their parameters. Parameters means del E, interaction energy, excess entropy minus T into del S, WFP, that means water finding probability, J value. Apart from WFP, we also calculated this J value above mentioned equations. So in this category 1, we have find that GMP substrate is basically enclaved or bonded by W3, 6, 1, 5, and 4 water molecules. This is only available in normal protein. Similarly, in HGMPR2, these water molecules we have find, they are enclave these positions. Okay, and category 3, either isoform 1 and 2, and category 4 in both cases. So what's its significance? What's its requirement? To calculate this energy, there is some implementation of computational drug discovery, because these water molecules is inaccessible in X-ray crystallographic structures. By MD simulation and implemented by inhomogeneous fluid solvation theory, I assumption and predicted these water sites and importance, because these water sites may help for water-based drug discovery. How is it possible? I show you in the next slide. That is the thing. That is the basic thing. These differences between X-ray crystallographic structures and MD structures. So what will be the differences? That means these water molecules W4, 5, 1, 6, and 13. These water molecules have specially EWW, that means in energy of water with bulk, and water with other conserved water molecules have significant lower value because they are in normal protein, non-cancerous protein. That's why they have very stable. But in category 2, in cancer protein, the same parameter, water energy and water with bulk energy is high. You can see also the value. Similarly, when we calculate the correlation coefficient of del E with entropy, we have found that the correlation is positive, 0.88 and 0.95. And here their correlation is very poor, minus 0.32 and 0.56. So in that cases, we are thinking the water molecules of category 1, that's mean normal protein, is also stable. So what is the implementation of these water molecules? So in category 1, these water molecules, W2, 3, 10, 8, and 17, these water molecules, we have find only the cancerous protein. In normal protein, this water molecule is absent. So we can utilize their coordinate, their geometry, and their electronic consequences for water-based drug discovery techniques. So the unique interaction of GMP N1, W2, SP129, that means this one, W2, SP129. This is one type of interaction, and another for W10. W10 is also this position. So this interaction, this one with this one. This is the two things, W2 and W10. We can utilize the position of water molecules, W10 and W2, so that the position will occupy some chemical group of the ligand. Therefore, what we do, we take the CH2NH2 group. Okay, to replace this CH2NH2 group, if we add this position CH2NH2 group, so that the NH2 group will occupy the position of W2 and it will be attached to these positions. Okay, so that is the thing. So incorporating the CH2NH2 group of GMP or NMP by affecting the binding affinity, increasing their solubility, and also the hydration susceptibility. But we'll consider only W2, not W10, because W2, if we check here, their interaction energy J minus 1.53 and here 1.58. So it is more stable. W10 is more stable and W2 is less stable. Okay, so it's very difficult, it's very easy to displace these water molecules W2 rather than W10 because that is more highly stable. Okay, so that is the importance of J value and these parameters. So for displacement of water molecules, W2 should be considered, and we also check their QSR property and ADMT test by adding the CH2NH2 group to this position. If we add this position, it will definitely replace this W2 position and it may be act as a good inhibitor for HGMPR2 activity. Okay, so the entire mechanism is like that. The entire mechanism depends on water molecules movement and displacement from substrate to product bound state. So initially W1 is entered, then W9, W13 is entered, then W19. So W1, W9 and W19, they are entered to this position. And which water molecule will left? Initially W4, 5 and 13, W1 to W14 and W9 to W13. These water molecules are left. In compared to the cancerous protein, the more number of water molecule enter and less number of water molecules exit. So that is also most interesting phenomena between HGM pair 1 and 2 structures. We have also calculated the volume of the substrate and product binding pocket. Okay, so during the simulation, we observed the substrate to product binding pocket, the normal protein is the volume is slightly different, like 798, and here is also 873. So their volume is increasing randomly, or more expansion is high compared to normal protein. So that is the most interesting phenomena. So this work we have published in Journal of Physical Chemistry 2021. The next work is water-based drug discovery in COVID, EMPRO protein. As we know, EMPRO is most interesting for COVID disease. So these are the background, which I am not describing in details. We'll consider the hydroxychloroquine. As we know, hydroxychloroquine may or may not be used for initial cases of COVID patient. So why not hydroxychloroquine? So I am thinking, if hydroxychloroquine is not used, so what will be its structural analog or repurpose of the drug? Or we can modify the hydroxychloroquine by adding some new group, so that it may act as a good inhibitor. So that is the main objective of my work. So these are the methodology. We have taken so many structures, like 10 to 12 structure we have obtained, and we have parameterized them properly. Okay. We also identify the water molecules of the crystal structures, and also the normalized B factor of each water molecules, because normalization of the B factor of water molecule is really necessary for checking the each water molecular position, thermal B factor or their fluctuations like that. So in the cases of 2, I present here the 3 features A, B and C. Initially, this mechanism I have predicted here. The two histidine, basically histidine 41 and histidine 163, and in middle part, cysteine 145 is there. So what is the main reason for not to discover for drug COVID? That is the mechanism based, because this is the dual catalytic mechanism. If we design some drug and which will prevent the biochemical activity of histidine 41, then cysteine move towards the another catalytic region 163, and it will go for its process. So it's very difficult for discovery of new drug. So what will be happen? We have rethinking and redesign some new drug, so that catalytic histidine 141 and catalytic histidine 163, both the structures or both the catalytic role of histidine should be prevented or should be inhibited properly. So that is the main reason for not to discover a drug for COVID disease. So this is the thing. Some Germany group, they already started this work, and they also crystallized one PDB structures. They designed one drug O6K, that is inhibitors, not drug. These inhibitors both attach to histidine 163 as well as histidine 41. So as the prevention, the catalytic mechanism of both the histidine. So their work is going on like that. So what we have done, we take the hydroxychloroquine. We also predicted the virtual screening method. The Swiss similarity program have been used, and several drug was obtained from my student from different databases. These are the final compounds which we thinking they may act as a good drug or repurpose of the drug for COVID disease. Hydroxychloroquine, chloroquine, methyl geometric like this, indacaterol. So these all drug which their similarity score to hydroxychloroquine is very good. So within this compound, we can rethink about the drug of COVID. So what we have done, we calculated their physiochemical property and toxicity property. The drug we have here, indacaterol, DB055039. These are the drugs which we observed their drug likeness score is 2.12 and drug score is 0.66 and hydroxychloroquine is 0.48. So indacaterol may be used and another for zinc here 0.50. So finally we obtain these zinc molecules which code is zinc like that 440 last three letter digit. Though its drug score is 0.50 than indacaterol, but in our computational results they are suggesting zinc will be good drug or maybe used for inhibitor rather than indacaterols. We also check the toxicity property of each predicted repurposed drug. This is the molecular docking techniques we have used by FEDBQT program and also followed by Autodoc Vina. This is the simulation methods. As we know, solvation, then energy minimization of solvated molecules. We have performed the energy minimization into 4 steps. Number 1, minimization of water molecules and ion. Then minimization the entire system by fixing the backbone of protein atoms. Then minimization with restraint of C alpha atom, then all atom energy minimization. So that is the gradually minimization process we have followed. Then heating from 0 to 310 kelvins up to 60 picosecond. Then equilibrium state, NPT, NVT, and NPT. And then finally production run. We have done the MD simulations of all these structures. So we take the native structure, pro-native, hydroxychloroquine bound structure, indacaterol, and zinc bound structure. So 4 conformation or 4 protein ligand complex we have created, 1 for native and 3 protein ligand complexes. And we also checking their RMSF value like that, they are fluctuating like this type. And also the RMSD value of this 4 type protein, MPRO, hydroxychloroquine, indacaterol, and zinc. This one, we have run some simulation of 100 nanosecond and 50 to 100 nanosecond I am plotting in this manner. Analysis of pocket volume. The POGMI program have been used to calculate the volume of each ligand bound conformation of the pockets. Similarly, protein ligand binding energy we have calculated by linear introduction energy method. Generally, the alpha and beta, these 2 parameters have been used, and the VDW and electrostatic, these 2 parameters have been used rather than used MMPBSA or GBSA method, because we already try MMPBSA and GBSA method, but the results is unrealistic data. So that's why I am thinking LIE is the best method and its data is very close to the experimental value. So this is our results analysis. When we perform the cluster analysis of water in the crystal structures, so this is the catalytic region, both histidine and middle is cysteine is there. 7, which is just far away from the histidine 41, this is the catalytic partner. So basically the imidazole group of histidine 41, the plane of the imidazole group and its stability will be maintained by 3 components. Number 1, aspartic acid, OD1, sulfur group of cysteine 144, and also these water molecules. These W1 water molecules, I have found almost all X-ray structures. They also maintain their stereochemical parameter of the imidazole group, and they also act, W1 also act as the catalytic partner. Okay, and this is the W2 is another water molecules. So normalized B factor, their normalization and Sasa value is greater than 0. In other conformations, if we take the suppose 20 crystal structures, we have obtained the results in two categories, category 1 and category 2. In category 2, we observed this histidine is far away from cysteine, and it will come and bind to this position of D187 with another water molecules W3. When we superimpose the A conformation and B, we observe W1, W2, and W3. They are also different positions. These are the table of PDBID water molecules, their normalized B factor, Sasa value, and their different type of Matthews coefficient, solvent content like that, extracrystallographic parameters. Binding mode analysis. This binding mode analysis we have performed by Autodoc Vina program. This is also checking the good fit methods. This is ligand profile interactions. Okay, so this is the comparative analysis between the interaction of molecular docking method and MD simulation method. In the upper panel, which indicates the molecular docking methods and its interaction. In lower panel, the MD simulations. If we check in hydroxychloroquine bound positions, this is the hydroxychloroquine and cysteine position is there. That del G is minus 5.8. And when we superimpose this hydroxychloroquine with the crystal structures, we find these W2 water molecules. Which one W2? This W2. This W2. Within this position, this W2 position is nearby the catalytic histidine. It's HCQ position like that. This is HCQ. So W2 and HCQ C11 position, their distances is within 1.8. Fine. When we perform the MD simulation of HCQ bound complexes, HCQ reoriented its conformations, and we have find C11. This C11 already occupy the position of CW1 because C11 oriented its conformations and take the position of W1 1.22. So docking result and MD simulation results will almost okay, but their binding energy, if we check, docking binding energy minus 5.8, LIAD minus 9.76, and MMPBSA minus 21, which is far very wide range. LIED and MMPBSA. That's why LIED is more acceptable. I take this value LIED. Okay. Next is indacaterol. Indacaterol in the docking study we observed they just replace the water molecules W2 because C22 atom of indacaterol they are within 1.8 angstrom. So it definitely replaces the position. But during simulation we find this indacaterol they are far away from the catalytic histidine, and they never replace any water positions. So possibly they induce the catalytic mechanism by water-mediated interaction. And we also check their binding energy minus 86 in docking, and LIED is minus 43, which is very good. And also MMPBSA is minus 10. Okay. Similarly, the zinc, which I support the zinc, this zinc one in molecular docking technique, we observe that C2 and O11 position of zinc, they replace. Okay, these two water molecules have been replaced by zinc 440. During simulation, they are also far away from W1 to W2 position, and they direct incorporate the cysteine and histidine region. This is one is crystal structure, another for is MD structures. And LIED method, we also check their binding energy minus 22. So I just ignore the Autodoc results, taking only MD simulation and LIED docking result. So if we compare minus 9.76 in HCQ, LIED method in indacaterol minus 43 and minus 22. Okay, so indacaterol may be used or zinc may be used for substitute of hydroxychloroquine. That is the thing. Here I am presenting the hydroxychloroquine, indacaterol and zinc bound conformation, how the protein is changing its conformations and the ligand from 46 to 50 nanosecond dynamics. This is the dynamical feature. You can observe hydroxychloroquine with histidine 41 and cysteine 145 and their movement. This one is the indicatrol. Initially, indicatrol is bind to this position, then they are far away. See the results. Yes, they are far away from the sites during MD simulations. Okay. This one is the zinc. Zinc is basically bind to histidine 41 and also the catalytic cysteine 145. In this way, they will maybe inhibit the catalytic process of MPRO by histidine 41 and cysteine 145. This is the structural, dynamical, and thermodynamic profiling parameters. We also again calculated the 4 parameter of energy of each water molecules and try to provide some biochemical mechanism. What's the mechanism of water molecules? A is the native conformation. In native conformation, this is the catalytic zone, histidine and aspartic acid, water molecule is there. Okay, so this native conformation to 3 ligand bound conformation, native to HCQ, native to zinc, and native to indicatorol. So all these cases, when we studied the results of MD, we find in HCQ bound conformations, W3 is departed, and the binding energy of W3, basically J you can check, minus 1.95. Fine. Then indicatorol also inhibit the histidine by the following path: W6, W4, and W5. In this way, they will prevent the or inactivate the systems. Okay. We also calculated the J interaction energy minus 0.95. Similarly, in zinc, they directly attach to this position, and interestingly, this histidine and cysteine may inactivate through the water molecule W7, and their interaction energy is minus 43. So that is the basic biochemical process if it in silico study. We also calculated the volume from native conformation to ligand bound conformation, means hydroxychloroquine, indicatorol, and zinc bound state. These are the comparative study of LIED method and MMPBSPB method. We suggest LIED is the best methods for predicting the binding energy, because this here in 49 minus 30.86 and minus 27.17, this is very good data, and they are very close to the experimental value. Okay. This is our conclusion. The conclusion indicate that zinc 28706440 may be act as a good substitute of hydroxychloroquine. So it may be further investigated. It may be used for experimental study for MPRO disease. This is my recent achievement. One book is published from Eliva Press. Two paper is published, one for Journal of Physical Chemistry B and Protein Structure Function Bioinformatics, and also another paper from Molecular Modeling and Graphics. This is their first page of the journals. Okay. Recently from MacOut, I have published these international books from Eliva Press with my MTech students, Urchita and Saborno. The work is published in Eliva Press entitled New Structural Insights of Human GMP Synthetase Enzyme, Computational Study for Drug Repurpose. Basically, the Department of Bioinformatics have following courses: MTech in Bioinformatics, MSc in Bioinformatics, and BSc in Bioinformatics. PhD Bioinformatics is already started. Few area of research is artificial intelligence, computational drug discovery, NGS, like that. This is my strength basically. Dipesh Chakraborty, who is working as a PhD research scholar, Urchita, Saborno as the MTech thesis student, Anushka and Shagata, they are from MSc molecular biology student. They did their master thesis under my guidance. I am very much thankful to my alumni, Alvia, Gyan, and Saima, who presently in PhD in different research institute and university. And from my group also, Devopriyo just recently joined as a project associate at Thasti, New Delhi. So if any query, please inform me. This is my phone number is there. Thank you. If any question, just inform me. Okay. Okay. That's very good. How many initial water molecules you choose before simulation? Before simulation is very difficult to choose. In my first work, GMP reductase, very few water molecule is there. Only 20 to 30 water molecules. So only 1 to 2 water molecules was there. So that is unpredicted. That is basically I have nothing any idea how many water molecule is there. So that is de novo based water modeling. That's why we perform all atom MD simulation and then predicted inhomogeneous fluid solution, predict their structures within the ligand, and then implemented in drug discovery. In second part, in X-ray structures, I predicted only 3 water molecules which are very close to the active site of the pockets. Okay, so and we also calculated their B factor, SASA and other parameters. And how to know the reactivity of compounds which not is used. Reactivity of compounds I can't understand. Can you explain it? Or any question? Reactivity of the compound is basically from some papers I am getting this information, the reactivity. Okay, and one thing and another, this substrate I know this substrate will come and bind to this position, that will be product or this is the intermediate step, like that. Next, whether it will be activated for reaction. Yes, it will definitely reactivate the enzymatic mechanism. For this study, we have to perform the QMMN calculations to check how it will be activated the enzymatic reaction or not. Okay, because my interest is what activation and inactivation of the water molecules, not like that. Implementation of the water geometry for structure-based drug discovery. Okay, so in this way, I am thinking like that. Or any question? Yes.
Yes, I'm here. Okay, thank you, Eshna. Thank you, sir, for the wonderful presentation. Good afternoon, everyone. So today was the last session and the final day of this 1 week online faculty development program on computational approach to molecular docking and drug designing and its scope in the entrepreneur world. At first, I, on behalf of the Department of Biotechnology, in association with IQAC, would once again like to thank all the eminent speakers who not only shared their valuable time and knowledge, but also gave us a clear understanding by providing the participants with a hands-on session that helped us to know about the various softwares and servers available and how to work with it. Now, I would like to thank all the respected faculty members for participating in this program. I hope you all had a good time with us. If you have any suggestions or comments, kindly let us know through the feedback system that we will be providing you shortly. Now, coming to the organizing committee part, I would like to thank our chief patron, Dr. Lakshman Chandra Sheth, Chairman iCare Haldia, uh, Professor and Dr. Shubhrama Gupta, the chairperson and head of the Department of Biotechnology, Haldia Institute of Technology. My sincere thanks to the conveners of the workshop, Dr. Sudip Dash, Associate Professor, Department of Biotechnology, and Dr. Mukesh Singh, Associate Professor, Department of Biotechnology, for their support throughout this workshop. I would also like to thank Ms. Eshna Dash, our beloved student, for coordinating the workshop so efficiently. And last but not the least, I would like to thank all the members who were directly or indirectly associated for their immense support for organizing this FDP program. So once again, thank you. And by this, I would like to conclude the program. And if you have any queries, any questions, do contact us, mail us, or you can even call or WhatsApp. So thank you.